Welcome to this lecture on evolutionary biology. Today, we are going to explore a fascinating topic, artificial selection. Have you ever wondered how we got so many different breeds of dogs? Or how the fruits and vegetables in our supermarkets became so large and delicious? The answer lies in the power of human choice. By the end of this video, you will understand not just the concept, but the exact mathematics behind how we shape the genetic destiny of species. Now that we have introduced the topic, let us define what artificial selection actually is. In evolutionary biology, artificial selection is the intentional reproduction of individuals in a population that possess specific, desirable traits. You'll often hear it referred to as selective breeding. Unlike natural selection, where environmental pressures like predators or climate determine which traits help an organism survive, artificial selection is entirely driven by human preference. We act as the selective agent. We deliberately choose the parents of the next generation to amplify a specific characteristic, deciding exactly who gets to pass on their traits. To truly grasp this concept, let us look at a classic historical example. Imagine you are a farmer thousands of years ago, looking at a field of wild mustard plants. You notice that some plants have slightly larger leaves than others. If you decide to only save and plant the seeds from those specific larger-leafed plants, the next generation will have even larger leaves on average. By repeating this simple process for hundreds of years, that humble wild mustard plant eventually transformed into the diverse vegetables we know today, such as cabbage, broccoli, and kale. This incredible transformation is not limited to plants. It applies just as powerfully to the animal kingdom. The exact same process of selective breeding is what turned fierce wild wolves into the incredible variety of dogs we see today, from tiny, friendly pugs to giant Great Danes. Instead of nature deciding which animals survive based on fitness, humans step in. We make the choice based on what we find useful, helpful, or even just cute. We are essentially acting as the ultimate matchmakers, pairing up individuals to bring out the exact traits we desire. Understanding this human-driven process leads us to a monumental realization in the history of science. Because the underlying genetic mechanisms of artificial selection are exactly the same as natural selection, Charles Darwin actually used it as a primary piece of evidence to explain his theory of evolution. Darwin observed how quickly and dramatically humans could change a domesticated species just by choosing who reproduces. He realized that if humans could cause such massive changes in a few generations, nature could easily do the same thing over much longer periods of time through environmental pressures. Before we move on to the mathematics, it is crucial to understand why this matters today. The concept of artificial selection is absolutely foundational to our modern way of life. Without it, we would not have the massive, high-yield crops that feed billions of people, like modern corn, which was bred from a tiny wild grass called teosinte. Beyond agriculture, it is vital in medical research, where scientists breed specific strains of mice to study genetic diseases. It also drives animal husbandry, allowing us to breed sheep for thicker wool or horses for greater speed. Now that we understand the profound impact of artificial selection, let us look at how evolutionary biologists mathematically predict these changes. This brings us to a famous formula called the breeder's equation. The formula is R equals h² times S. Let us break down these symbols. R is the response to selection, which measures how much the offspring's average trait differs from the original population. The symbol h squared is the narrow sense heritability, a value between 0 and 1 indicating how much of the trait is passed down genetically. Finally, S is the selection differential, representing how much the chosen parents differ from the original flock. If you increase the selection differential by picking more extreme parents, the response R will increase proportionally. Let us see this formula in action with a concrete example. Suppose you are a farmer with a flock of chickens averaging 2.0 kilograms. You want heavier chickens, so you select parents that average 2.5 kilograms. The heritability is known to be 0.6. First, we find the selection differential, S, by subtracting the original average from the parent's average, giving us 0.5 kilograms. Next, we multiply this S by the heritability of 0.6 to find R, the response to selection, which is 0.3 kilograms. Finally, we add this response to our original average. The new generation will average 2.3 kilograms. We have successfully increased the flock's weight in just one generation. Having successfully used the breeder's equation to predict offspring traits, we sometimes face the opposite problem in the real world. What if we already ran the breeding experiment and want to know how much of the trait was actually genetic? We start with our standard equation, R equals h squared times S. In this scenario, we can measure the selection differential, S, from our chosen parents, and we can measure the actual response, R, from the offspring that grow. The unknown variable is the heritability, h squared. To find it, we need to isolate h squared algebraically. This setup is crucial for scientists trying to understand the genetic limits of a specific population. Let us complete this derivation to find our final formula. We begin with R equals h squared times S. To isolate the heritability, we simply divide both sides of the equation by the selection differential, S. The S terms on the right side cancel out perfectly. This leaves us with our final result, h squared equals R divided by S. This beautifully simple rearrangement is incredibly powerful. It tells us that the heritability of a trait is simply the ratio of the actual response we got divided by the selection pressure we applied. Now that we have proven this relationship, let us apply it to a real agricultural experiment. Now that we have our rearranged formula, let us look at an agricultural scientist trying to increase oil content in corn. The original corn averages 4.0% oil. The selected parents average 6.0%, and the resulting offspring average 4.5%. First, we find S by subtracting 4 from 6, giving a selection differential of 2.0%. Next, we find R by subtracting the original 4% from the new 4.5%, giving a response of 0.5%. Finally, we divide R by S. 0.5 divided by 2 gives 0.25. This means only 25% of the variation in oil content is genetic. To truly cement our understanding, let us visualize this entire process on a graph. On the screen, you see a bell curve representing our original population, with the trait value on the horizontal axis and frequency on the vertical axis. Notice the shaded region on the far right. These are the extreme individuals we selected to be parents. The distance from the original population's center to the center of these selected parents is our selection differential, S. Now, look at the dashed green curve representing the next generation. It has shifted to the right. The distance it moved from the original center is our response to selection, R.
Because heritability is rarely perfect, the green curve does not move all the way to the parent's average, but it clearly demonstrates evolutionary change.
We have covered a lot of ground today, so let us summarize the key takeaways. First, artificial selection is a powerful process driven entirely by intentional human choices, allowing us to shape the genetic destiny of species. Second, this mechanism is absolutely foundational to our modern world, giving us high-yield crops and diverse domesticated animals. Third, we learned that this is not just a qualitative concept. The breeder's equation allows us to mathematically predict and measure these changes with precision. Finally, artificial selection proves that evolution is not just a historical idea, but an active, measurable process that empowers us to innovate for the future. Great job following along, and keep exploring the wonders of biology.